Hi everyone. I'm going to go through an actual annotation of a Drosophila species Contig feature, and I'm going to use this to enable you to see how you could navigate through these different databases to annotate your feature. So there are certain sites that we'll be using a lot. So I have these sites open right now. These we have the Genomics Education Partnership site, gep.wustl.edu. I also have the UCSC Genome Browser site, and I have Flybase open and also Blast, NCBI Blast. Now, in order to get to the UCSC Genome Browser gateway, I go to Genomics Education Partnership, the GEP site, go to Projects, and under Projects, I can click on the GEP UCSC Browser Mirror. That'll take me to the gateway. When I get to the gateway, I could search for the species that I'm actually looking for. Now, the species will change as uh, throughout the years, but in this example here, this is just a practice example of one of the features that I annotated some time ago. This is from Drosophila ugracilis. So typing it in the search box, you could actually bring that information up. So that's the species that I'm searching for, and that the year is actually 2017. And I'm annotating the control for that year. So the Contig that I'm actually gonna annotate is Contig 62. So I could put in Contig 62 in that box and press go, and it'll take me to the alignment page where we have color diagrams, where we actually have the possibilities of these, these different databases here, comparing it to Drosophila melanogaster to see which ones could makes the best comparison visually for Contig 62. So clicking on these databases here, GenScan, GenID genes, NScan, GEP genes, etc. These could all be used to compare to the Drosophila melanogaster above here. But only interested for this search, I'm only interested in what we're going to look at for GenScan. So I'm going to annotate the feature, one of the features on GenScan up here. How do I know which is a specific feature? Well, first of all, let's just get rid of the other databases, and I'm going to condense the Drosophila melanogaster genes, so this could be a little bit clearer to see. All right, so condensing Drosophila melanogaster genes, make it dense, and I'm going to hide everything else except GenScan, because this is where I'm going to get the information to annotate the gene. So I hide everything else, and I'm going to refresh. So right now, all we have is Drosophila melanogaster above and GenScan below. Here, I left the RNA mixed embryo RNA information. I mean, this is also important to see where there might be or might not be genetic information based on the example that we see above here. Okay, in order for me to see which feature is actually going to be highlighted here, by just clicking on any one of those exons or features, it's going to refresh the page and it's going to actually number the features for us. So this here is feature 1, feature 2, this is feature 3, this is feature 4, feature 5, etc. Right? Now I'm interested in annotating 62 Contig 62.3. So that's 62 Contig 62 feature 3. So this is the one I'm going to get the information from. So in order to get protein sequence information, amino acid information, I could click on this feature again, and it's going to bring me to this page, the GenScan gene predictions. And here under links to sequences, I'm going to click on predicted protein. Predicted protein here gives me the amino acid information. So highlighting this and bringing it over to Flybase, Flybase will do a BLAST. In this case here, we're going to annotate the proteins, right? And Flybase will do, we could paste that information in here, and Flybase is going to do a BLAST P for our annotated protein. Now make sure your default, the species is Drosophila melanogaster, and if we blast, this might take a while, but blasting would help us to see the alignment of the protein information, which we got from GenScan, right, for Contig 62.3, and it is going to blast it against Drosophila melanogaster. Now, the Drosophila gene that is highlighted, or that we're comparing with, is CG14456. So copying this and bringing it over, because we now want, because this is the gene that is highlighted in Drosophila melanogaster when we compare it to our predicted protein from Ugrasili 62.3. So in order to find out additional information about that predicted protein, or sorry, that gene from Drosophila melanogaster, we could go to the gene record finder and paste that gene into gene record finder, and finding record will give us here not only alignment information, it gives us the CDS usage map. And in this case here, the CDS usage map has 2 isoforms for our specific gene. And we have 3 to 4 exons. So our isoform is on this vertical axis here, and on our horizontal axis, we could actually see our exons. So I'm going to start with annotating my first exon on this. But before I even do that, let me show you something. Here, let's highlight this a little bit by clicking on it or zooming in ×3. Going to feature 3, scrolling to go to feature 3, I just want to show you what it looks like. Here we see a possibility of 1, 2, 3 exons. And going back to the gene record finder, we see 3 exons and possibly 4 exons. This first isoform only has 3 exons, while the bottom one also has 3 exons. So in this case here, the third exon and the fourth possible exon here might be one and the same, right? But we will see that as we continue with our annotation. In order to get this protein information from this gene here, we could just click on a specific exon, and the protein or amino acid information would come up, right? So I would copy this amino acid information, and I would go to BLAST, and I would use BLAST X, because we're looking at we're going to look at a specific nucleotide query from Drosophila ugrasilis 62.3. Right, we're going to look at the nucleotide query here. So I'm going to upload the file. So now you could do one of 2 things. You could either upload the Contig 62 file from which will be available to you on Blackboard, or you're going to upload your specific Contig file. That Contig file, or this it has sequence information with all the DNA information inside of it. So this is one way you could get that information. Another way you could get that information is going to the UCSC genome browser site, and where we have Contig 62, go to View, go to DNA, and we could get DNA. That gives you all the DNA for Contig 62, right? But I chose to upload it from the site. It'll give you the same information. So remember, we copied this nucleotide information here. So we're going to compare our nucleotide query information in this first box up here, and we're going to align 2 sequences, and we're going to compare our nucleotide query to our actual protein sequence, or our amino acid sequence. So scrolling down to adjust our parameters, we're going to make the word size, or this should be a default of 3. If it's not, you should put the word size on 3. Get rid of the low complexity regions and have no adjustments, right, for compositional adjustments. And then we just show results in a new window, and we blast that. Now blasting that might take a while, but in our case it uploaded pretty quickly. So this is the first piece of information that we're going to actually use. This is very, very important. So here we see alignment of our nucleotide sequence, or our query and our subject, right? And the query information is going to give us the possible coordinates for the first exon in our Drosophila eugracilis. The possible coordinates is starting from 2685 and it's going to end at 2750. The frame is also very important, so we're going to make note of the frame. So what I did is I opened up a Word document. So everything you write, you do will be documented. It makes it easier to follow. So I'm annotating Contig 62.3. Now the details of what you write is going to depend on you and how well you could follow what you write. So for exon 1, which we looked at here, we have a frame of 3. And the coordinates that we use is 2685 and it goes to 2750. So what I will do is I will go to this genome browser site and I will click on this box here. And for my Contig 62, I'm going to put my coordinates 2685 to 2750. Just going to paste it in here and go. So this is my exon, but it makes sense. We can't see the entire exon. So in order to see the entire exon, I'm going to decrease my coordinates here, 2685. I'm just going to put 2600. And 2750, I'm just going to put 2700. So we can see our entire exon in our field of view. That did not work out too well. Oh, here we go. Actually, it's 2750. So we have to increase. So it's 2800. So I decreased on one side so I could see the end of this exon and increased on the other side so I could see the other end. A lot of this is not necessarily intuitive, but you have to use your discretion and how you understand how to navigate through the database in order to get the best possible view and the best possible representation of what you're looking for every step, right? And as I go through this, I'm going to make a lot of mistakes as we go along here to show you that how possible it is to make certain mistakes, right? Even I make those mistakes as I do my annotation. But the goal is to continue to keep an open mind and continue to use the information that we know in order to navigate through the databases and to annotate our gene properly. So here's our entire exon. Drosophila melanogaster on top, GenScan prediction at the bottom, and this is Drosophila eugracilis. So this is one end here. In order to get the full representation so I could see my reading frames, what I would do is go to base position and I would make this full. And refreshing it make everything bigger, and I could actually see my frames. This is the positive frame. Notice the arrow is pointing in this direction here. That's the positive direction. If I click on my arrow, it's going to turn to the other side. That's the negative strand. So I want the positive strand because if you could see from our blast alignment from NCBI blast, we were looking at the positive frame. The positive is in positive 3. So that's why we're looking at the positive frame here. I'm going to highlight the beginning of my exon here to make it bigger, right? The reason why I highlight the beginning of my exon is because ultimately what I want is to have a beginning of my exon. I would have a start site here, right, for this first exon, because we see there's a possibility that there might be 3 exons, 1, 2, and 3, right, from this site here, right? We notice there's 1, 2, and 3 exons. Now there might be 4, but in this case here, we're looking at 3, a possibility of 3, right? So in order to annotate, we need a start site at this end here, and we need a stop site at this end here. Everything in between will be an acceptor or donor site, because remember the goal is to splice our introns out. So we could only have our exons, so we could see where the CDS is, or the coding sequence for our exons coming together, right? So we want to splice these introns out and splice our exons together. So we need a start site here. For our start site, we could go to this database here, and for this start site, you notice this methionine, ATG. Now, we want the information from the third reading frame, frame 3. That's what we see here, right? From the third reading frame here, right? Frame 3. So what we're going to do is go to this database, and ATG, we're going to look in frame 3. And of course, it shows a start site. Now the start site might not always fall here, right? You have to look around, use your discretion, use the frame, and use the necessary start site. In this case, it makes perfect sense. Methionine is right here. Methionine actually starts, I'm going to highlight this and make it even bigger. I'm going to highlight it, make it bigger, so we could see where this methionine starts. This methionine actually starts here at 2,684, right? So that is going to possibly be our first set of coordinates. 2,684 is where our methionine starts. Actually, ATG. So even though it spans here, our A starts here, TG. So actually it starts at 2,685. This is where our A is, right? This is where this starts at 2,685 at A, because this is ATG, our start codon, right? Because we have this ATG at 2,685, I'm going to make note of that, 2,685. This is where our start site starts, our exon starts for exon 1, right? So now, in order to find the end of this exon, what I will do is I'm going to scroll, right? But this is going to take forever because it's zoomed in. So I'll zoom out times 3 to make scrolling a little bit easier. Well, I'm going to zoom out to times 10 to make scrolling easier. And what I'm going to do is I am going to highlight this region here, the region that I'm going to use. I'm going to try and highlight this region, right? I'm going to highlight it some more so it'll be even bigger so that it's even more visible. So remember, now we just looked at the beginning and we found the start site at 2,685. What we want next is the splice site donor, because remember, the splice site donor comes at the end of an exon or the beginning of an intron, right? So a splice site donor we said was GT. The splice site donor was GT, so at the end of this first exon here, we look for a possible GT, right? And the possible GT, remember this is the positive frame that we're looking at, or the positive strand. So this is GT here. We're looking in this direction, GT. And anytime you annotate GT in the positive strand, you're going to look to the left of the GT, right? So to the left of the GT, and remember we're in frame 3. We're in frame 3 for exon number 1. We're in frame 3. So because we're in frame 3, this GT, we're going to look to the left, and we're going to look to frame 3. So we're going to cover this GT, right? And looking to the left of GT, right? The left in frame 3, the GT ends right here, right? Where there's V. The GT ends here. So because the GT ends here, the next in frame 3, because the GT end here, the phase that we're actually going to be looking for the beginning of our GT here, the phase that we're going to be looking for is at 2,750. So for frame 3, the phase is 0, but it's 2,750, right? So we have this 2,750 for the GT. So for every GT, for every donor, you need an acceptor site. So for our next exon, right? Because remember, exon 1, so we have from 2,685 here for our start site. We go through frame 3, right? We look through frame 3, and looking through frame 3, another thing that's really important is while we scrolled, you had to be looking at the bottom here through frame 3 to make sure that there is no stop codon. If there is any stop codon or any red in frame 3 as you scroll, right, you would not use frame 3, and then you would go to NCBI BLAST and you would use the other alignment and you would start the process over, because what we want to do is eliminate any possible stop codons prematurely within our exon. But in this case here, there was no stop codons, right, when we scroll. So since we have the start and the stop, we have the start site here, and we have the donor site, we need an acceptor site for exon 2. Right? In order to get an acceptor site for exon 2, you have to go back to the gene record finder to find nucleotide information for that exon. So I'm going to copy that exon nucleotide information, I'm going to go back. So remember, you have to take an image of this. I'm going to shut this down so I don't get confused. And I'm going to go back to NCBI BLAST, and I'm going to paste the nucleotide information here. Up here, we already have this gene, the nucleotide, the DNA for Contig 62. So we're going to just do exon 2 now and blast that. Parameters will stay the same as before. And when we blast it, we got this page with a fresh new alignment. This alignment looks really good. First of all, because we have a 98%, 93% identity, we have no gaps. So this alignment looks pretty good. We're using frame 2 here, right? So it starts at 2,810. Our query starts at 2,810 for exon 2, 2,810, right? And it's going to end at 3,124. So that is the coordinate. These are the coordinates that we put in here. 2,810, 810, 2, 3,000, 3,124. Right? And we're going to blast that, and we're going to search this. Remember, again, it does not show us our entire exon, so I'm going to decrease this to increase this side to 3,150, and decrease this side of my exon to 2,800. And in this case here, we're going to see our entire exon. But what we want is this side, because remember, we want the splice site acceptor, because we already have a donor from exon 1. So we need this side here, because the exons are going to come together in this way. All right, so we want that side of the exon. So this is the side we're going to look for. Right? So in this case here, because we're dealing with exon 2, we're not looking for a start site, we're looking for an AT site, or an AG site, sorry. We're looking for an AG site. So I'm going to scroll. Well, we find my first AG site. No AG up here, but I see one. Let's see. This might take a while. AG. There's an AG start site right here. Right now, there might be multiple others, but this is the one that I'm going to use here right now. And you might have to test more than one, right? So this AG site here, remember, for exon 2, we're looking at frame 2 for this alignment for exon 2. Now we're looking at frame 2, and our AG site is here, right? So this is frame 2, and it ends here, right? This AG ends here. So we're looking to the right. A while ago, we looked to the left of the splice site donor. We're looking to the right of the splice site acceptor, AG. We're looking to the right of the splice site acceptor on our positive strand, right? Frame 2, looking to the right, the next amino acid starts where? At a phase of 0, right? Now, this at a phase of 0 is where our next amino acid starts. There is nothing, it ends, the AG ends right here, so there is nothing, no letter between the AG and the next amino acid in frame 2. So this is a phase of 0. Remember, a 0 could match up with a 0, so we had a splice site donor, the phase of 0 before, and now we have a splice site acceptor with a phase of 0 also. Right? So that could match up, because remember, 3 matches with 0, 0 match with 0, 2 match with 1, right, for splice site donor and splice site acceptor, right? So we have a phase of 0, so now all we have to do is write the coordinates for where we got our AG phase of 0 from. And our coordinates is 2,810. This is where the next letter would start, 2,810. Right? Phase of 0, 2,810. Right? So now that we have this phase 2 here, and we documented where AG is 2,810 here, what we're going to do now is scroll and make sure there's no stop codon within our frame 2. I'm going to decrease this again, zoom out, so it makes scrolling easier. There's no stop codon yet, none. No stop codon in frame 2. We see one in frame 3, but it's not in frame 2. And it doesn't matter if you see a start site, it's just going to code for a different amino acid. That's fine. Scrolling through it, still no stop codon. right? I'm going to scroll through the entire thing. It makes sense to look at every single possibility until I get to the end of my exon 2. Still in frame 2, right? Because still in frame 2, at the end of my exon 2, I am going to find a GT site, because you want now, we had start, donor, we had acceptor, now we need a donor site for here, that is GT. Right? We need a donor, which is GT. Now there are a lot of GTs here. We have one here, we have one here, and we have one here. Right? So the GT, after I tested all of these GTs, now you have to meticulously test each one of them to see which one, to find the phase of every one of them for frame 2, to see which one could match up with the phase of the next one of the exon 3. Okay? So we have to do, what I did was I found GT, the first GT that we see here, looking to the left of that GT, in frame 2, we have, actually this is the first GT that I found. This is the first GT here, looking to the left in this frame 2, it's the phase of 0, right? So phase 0, 3134, GT, phase of 0, GT, phases, because it ends here. So it's 3134. This nucleotide here is 3000, GT. This nucleotide here is 3134. All right? So this 3134 is phase 0. The next GT is here. I look to the left of that GT. So we have, actually when you look to the left in frame 2, it ends here. So there's a nucleotide here. This is phase of 1. This is a phase of 1. And our phase of 1 for this GT here is 3000. If this is 3130, this is 3129, this is 3128. This is 3128. 3000 actually 128. GT ends here. 3128. Fine. The next GT is here. And actually this 3128 we said was in frame 1. We already said that. Phase 1, sorry, in frame 2. GT, the last GT that we saw in this frame, in our reading frame here, is this GT here. And that GT looking to the left of it is again in phase 0 because our V ends here from frame 2. It's phase 0. So it's phase 0 and GT, this phase 0 here, this is the letter we're looking for. This is 3124, right? So this is 3124, and we're going to use that here for our splice site donor, 3124 for exon 2. So now all we need is exon 3, the nucleotide information for exon 3. We need a splice site acceptor for exon 3. So in order to get that, we'd go back to Gene Record Finder, right? And we'd look for the information for exon 3, the amino acid information, and go back to BlastX. Now I'm going to close this page because I already have all this information here from each one of the alignments. And I'm going to blast that. Again, I'm going to leave this Contig 62 DNA here, and I'm going to just blast. And blasting it takes me to another alignment page for exon 3. This is in frame 2. And it starts at 3188, and it ends at 3448. So I take these coordinates for exon 3, 3188, and I go to 3448. And 3188 to 3148. Double check. 3188, 3448. 448. And again, remember what we have to do is we have to increase this side. So I'm going to increase it to a certain number, and I'm going to decrease this side so I could see my entire exon. Right? And this is my entire exon 3. Now, all we needed was a splice site acceptor on this side, right? I'm going to zoom in. Why we need a splice site acceptor on this side? Because of what we need here. Because we had a splice site donor for exon 2. So now we need a splice site acceptor for exon 3. And zooming in even more, I'm going to see more information. And the frame that we were in before is in frame positive 2, is in frame 2. And because we're in frame 2 here, we're going to look for our specific coordinates where it said that we started, and where it said that we started for exon 3 is 3,188. 3,188 is about here, right? So now we're looking for actually an AT site, right? We're looking for, an AG site. We're looking for an AG site for our exon 3. We're looking for an AG site for our exon 3. Scrolling. Our first AG site is not even at the, oh, it's right here actually. Sometimes it might not be at the beginning, it might be further left, it might be further right, but it's for us to find out, right? This is the only AG site that we see here in the frame. It makes sense. Now we're going to look for the right, we're going to look to the right of AG. Remember, we looked to the left of GT. We're going to look to the right of AG, right? And we're in frame positive 2. We're in frame 2, right? Actually, there's a stop codon here, but the stop codon is where our actual splice acceptor ends or begins. So this will be spliced out. This AG will be, this here in frame 2, this stop codon will be spliced out as part of the introns. So we don't need to pay much attention to that, right? Because it'll be spliced out. Looking to the right of this AG, it actually starts right here, right? AG starts right here. The next letter the next amino acid starts right there. This is a phase of 0, right? This is frame 2, phase 0. So you document that also. Frame 0, phase 2. Sorry, phase 0, frame 2, right? Phase 0, frame 2, right? So we're going to continue, right? And at the other, so we're going to continue because this actually makes sense because our phase of 0 matches with this here or it matches with that up here, right? It matches with this one or it matches with that one, right? From our exon 2 for our splice site donor, right? So we can get rid of this one here, right? Because it's phase 1, it doesn't make any sense. Get rid of that one, right? So we have one of two possibilities. So our splice site donor is 3,188 and it says to 3,448. So I'm going to scroll to see if we have any stop codons within frame 2. And I'm going to zoom out a little bit and to make scrolling easier and faster. Zoom out a little bit more, make scrolling even faster. And we have this end here, right? So as we looked through while scrolling, we didn't see any stop codon within frame 2, right? So we already matched the possibilities of exon 2 here. So we have a start and stop. We have a start in GT. We have a AG and GT, right? Two possibilities for this GT here. And we have one possibility for this AG here, right? And our one possibility is phase 0, and it's at 3,188. So now all we need to find is a stop codon here in frame, and we need to find a stop codon within frame 2. And we go all the way, we see the first stop codon within frame 2 is right here. So I'm going to highlight the spot to find the coordinates. Highlighting the spot to find the coordinate, you see the stop site is right here. Now, we took from the beginning of the start site, but we don't want we don't want the end of the stop site. We want the beginning before it actually stops. So as soon as it stops, that's the coordinate we want, right? And the coordinate is 3,445, right? It's 3,445. So that would be our stop site, 3,445. So putting it all together, we have coordinates, the start site, I'm going to highlight this yellow, 2,685. We have the next set of coordinates, so this start site, we have the end, right, is 2,750 for our first exon, right? Then for exon 2, we have 2,810 for our AG. And our GT, we have two possibilities here, 3,134 or 3,124, right? And for the AG, we have this 3,188 for exon 3, and our stop site we have 3,445. So these are the coordinates that we're going to use and put it into our gene model checker. Okay, so gene model checker. So we're done with the annotation of all our possible exons here, and let's look at this to see, because this is a possible. There's a possibility that it's possible that there are 4 exons. So clicking on this, highlighting this entire thing here, let's blast this also. Let's just see, and I'll show you something. So blasting that brings me to a page where it says that my coordinates are in frame 2, 3,233 to 3,412. Actually, 3,412 actually falls within, you notice 3,233 to 448. 3,448. This here falls within our range. 3,448, our exon actually ends at 3,445, and our actual feature ends at 3,445, and 3,233 is within the range of one of our exons. So actually exon 3 and the possibility of an exon 4 is the same thing. There are no 2 different exons, it's the same thing. It falls within that range. So now we could go to the gene model checker and put our coordinates in. How you do that is up here where it says Contig sequence file, you go to browse, and you're going to upload your specific Contig, the DNA for that entire Contig. I uploaded Contig 62. The ortholog name that we're looking at, that's in Drosophila melanogaster. Remember, we used from our gene record finder, we use CG14456. So we're going to go here, CG14456. All right, and then it's going to ask us which isoform we annotated. In our case here, we only annotated the top isoform, right? We annotate, if we want to annotate isoform, this isoform here, we could just click on that. But in this case, we're only annotating so far this top isoform. Now, when you're given a feature, you're going to annotate all the isoforms within that feature, right? In this case, this isoform here and that one is exactly the same, right? So I annotated this top isoform, so it's PB. So I'm going to put that in here also. PB, right? And here is where I put in my coordinates. So my coordinates started at 2,685 to 2,750, 2,685 to 2,750, right? So because I already have it written down, I'm just going to paste it in here. And it's separated by a comma and a space. The next set of coordinate is 2,810 to 3,224. 2,810, 3,224. Now, let's try 34 first, right? 2,810 to 3,134. 3,134, space, comma and space. To the last set of coordinates, which is absolute, 3,188, right, to 3,445. 3,188 to 3,445. And we did the positive strand, so we're looking at plus. Annotated untranslated region, no, we did not annotate the UTR. In this case here, complete gene model, yes, it's an entire feature that we annotated. That's all these coordinates are for an entire feature. So clicking on this box here, it automatically updates your stop coordinates according to what you put in here, right? And we annotated Drosophila eugracilis. You're going to look for it. In this case here, you're going to put the specific thing that you annotate, whether you're annotating a control or a dot chromosome. In this case here, I annotated a control, and my project name is Contig 62, all small case letters. Now, this here might be a capital letter, CG. So that's, yeah, it is capital letter. This might be case sensitive. And in here now, you just verify your model. And it tells you how many here you passed and you failed, whether you passed or you failed. In order to analyze this information here, you could actually click on the drop-down menu and it tells you where the problem is. And you can zoom in and it takes you back to the UCSC genome browser, and you could go back and look at your coordinates here to find out whether you put in the right coordinates. In our case, we had two possibilities for GT here. We used 3,134. Let's try 3,124 for this exon 2. So 3,124, right? And clicking this box again, it automatically updates and verify our model. You notice everything is passed now. So these are the coordinates that we need, right? These coordinates, that is basically the solution to what we're doing, our problem. We're finished with annotating that feature. If you're given one feature, you're done. If you're given more than one feature, you have to annotate the other features. Remember, if your feature has more than one isoform, you also annotate every single isoform as long as it's unique. Right? After this is all passed, you have passed everything, you could get the dot plot by clicking on dot plot. This transcript sequence, you could get the information for that transcript. Peptide sequence, extracted coding exons with all the CDS information here, the start and the end sites, and you could download the specific files that you might need to complete your report. Right? Everything is here in the checklist as passed, so we are all done. Remember, when you're doing this project, to make sure you take an image of every single step that you do for documentation, because you will need to upload it in your report. In addition to having a picture of everything, you should also document everything that you do in your lab notebook so you don't have to go back and forth. You document it in a similar manner, like I did here, with more, of course, writing so you could follow along. Right? So this is how you annotate an entire gene feature, well, a Contig feature, and I hope that helped you guys out, or it will help you guys out. Good luck.